for the haircut.
Oh.
Good.
Good. How are you?
Good. So, are you excited about today? Learning a new skill? People who liked ImageJ will like this one too. Or if you're creative, you would like this one too. This one is a fun one. I like it. So before I get started with today's stuff, do you guys have any questions about last week's assignment? Is anyone still having any trouble with any of the sources, not being able to find them, or any other issues like that? Cool. So if one of you, I do see one person saying that they still need some help. If you're still having some issues, please either stay back after we are done with today's session, or you can send me an email and I can set up a Zoom call with you to help you out, okay? Okay, I'm gonna go ahead and share my screen so we can look at our stuff for today. So today we are starting a new module within this library research skills, where we are actually going to take all this information that we've been finding, all the research that we've been doing, and we're going to synthesize it into our own schematic to show the mechanism of action for our drug, or how it would interact with the body in general, or specifically with the condition that you have started to look at. So if you go into that module, the first page obviously has the overview of what it is that we're going to be learning this week. So our objectives this week are to understand the 3 stages of signal transduction pathways. We've been going over that for several weeks now, right? So we've talked about how you have a signal that comes in, it talks to a receptor, or it binds to its effector, and then that's going to start the signal cascade by activating modulators that are then going to affect other proteins that will act as your final response, that will induce the final response. So that's basically the 3 stages that we are talking about. So we would want you to be able to identify them given a pathway or a schematic on a test or a quiz or a paper. The other thing is, obviously, we want you to be able to name and describe those different steps in the pathway itself. But then also after today, we would want you to identify them in a pathway analysis sense. So in an actual schematic, how is it that we organize these different parts into a cohesive story or a cohesive pathway? Today, we're going to talk about differences between what we call a node pathway versus a network when we are developing these schematics. And then we are going to make our own schematic at the end of the day that that's your assignment for this week, using either PowerPoint or Adobe Illustrator. Now, I will show you how to do it in a PowerPoint slide. And most of you, I think, will be very comfortable using that method. So Adobe Illustrator is only for those of you who maybe are already familiar with it and like using it. I've had some people that are interested in graphics or designing and want to use that, and they can use that by all means. But the way I will show you is using a PowerPoint. Okay, so within the module, you're going to find some links that you can go to get more information and learning about signaling pathways in general. So I've given you a site from NIH. I like this site because it describes biological pathways in a very simple, clean way. So it's very easy to understand, and it's written kind of like in a question-answer sense. So you can look at each one of those questions and see a very clean answer for them. So I highly encourage you to go through this to look at that basic information. And it talks about why is it important for us to understand as researcher how biological pathways work. It talks about how we find these pathways in a lab, in a wet lab, while we're doing our experiments, and then how we can relate them back to disease. So this is, like I said, a very cool, easy to understand site on signal transduction, biological pathways, things like that. I also have a PowerPoint that talks about why we would want to run pathway analysis like the one we are going to run today, and how it helps us understand, understand the significance of our data. All this data that all the researchers put up online, or that's published, it only contains like tiny little pieces of information for the biological pathways. Even when they are running whole genome analyses, it's still going to be a very small part of the story. So by running these powerful algorithms inside these computer programs and softwares that are now available, we can integrate multiple thousands of these studies that only contain a little tiny piece of the puzzle and make a whole cohesive conclusion out of it. We can get more information out of it than we can in just the little parts. And so this will kind of go through that, the importance of that, why we do these things, and how we go through pathway analysis. Underneath it, there are two separate videos. There are two videos from YouTube, and then there is one from me. The two videos that I have from YouTube, one is an introduction on the platform that we are going to use to run pathway analysis, which is Reactome. It's an open source program, so you can easily access it just through the web and work with it and get your results very easily without any need of additional sophisticated softwares or understanding algorithms or being able to write, algorithms or anything like that. When I've done gene expression array analysis in the past, or before these sophisticated softwares were available, I would actually sit there and write my own code in order to analyze it. Now, these softwares allow us to just do those same work. The same work can be done in hours or minutes that we used to take a month to do or weeks to do. So that's really powerful tool that we have available, completely free and accessible to everyone who wants to use it or try it. The second of the two videos is a video that is more a demonstration of how you can use and kind of an introduction on what different parts of the program show to you, the general layout, how to run a simple analysis, and things like that. However, even with that video, I felt like it may be hard to follow for some of the students. So I actually made my own little demonstration, which is just 12 minutes long, and it's catered to just our own assignment. So that's the one that you want to go to when you want to do the actual assignment, because it's going to focus simply on what it is that you are going to be focusing on and working on. And just, because we're going to use this platform for a very basic, very simple kind of analysis, not the more complicated, sophisticated ones that it's capable of. So this is just kind of going to take away all the noise, this particular video, and just show you exactly what it is that you need to do. And then once you've run those, your final assignment is to make a schematic about your story, about your problem and your research that you're doing, where you will make an original schematic of your own to put inside your poster. This is going to go eventually in that final poster you're going to build. That will show some genes and proteins that you found affected by either your condition or the supplement or both. And you're going to integrate them into a schematic to show how your natural product affects your condition or disease that you're focusing on and working with. Now, this is an example of what it is that you will be kind of doing at the end of the day. You will have information on the specific genes and proteins that you are going to be calling your nodes. Those are your individual nodes. These are the genes and proteins that you find affected by the natural product that you're focusing on. Once you run the pathway analysis, you're going to get information on large networks or the big, kind of pathways that are affected, big categories of pathways that are affected by your natural supplement. And those will be your networks. So for example, cell cycle is a network, DNA repair is the pathways are networks. So those would be your main networks. And then you will have the individual pathways that are affected, associated within each network by your compound or by your condition. Now, in this picture, this is just something that I want to clarify, and it's written here as a note as well, that in this picture, I've just provided you with images from online. What you are doing is not providing me with an online image, but rather making your own. So you are just taking the important portion of your pathway and making it your own. And we'll talk about this more in detail as we go through today's demonstration. So you're going to be making a single PowerPoint slide, which you will turn in. You can submit your final slide as a PDF to make sure all the, nothing changes in formatting, or you can put it in as a PowerPoint slide. I'm okay with either way. Yes, you will be this is a group assignment. You will be doing it as a group, and you will be turning it in as a group. And I will show you how you can use the tools inside PowerPoint to do these things, to make a schematic. However, for your information and just to help you out, there are a couple of tutorials listed underneath, YouTube tutorials on how we can create path diagrams in PowerPoint, how to make a flow chart in PowerPoint. So these are some of those things that you can use, tools that you can use to kind of learn some ways that you can enhance your PowerPoint skills and make different things in there. Okay, so let's get started. We're going to start off by what you need to start with in this software is, I'm going to first introduce you to the Reactome, then I will talk about that. So Reactome is an open source software or platform that we're going to use that contains, it's constantly updated. It contains all the pathways that are found up till now, and it contains all the individual proteins that are affected within each pathway and how they're connected to each other. It is also constantly updated with new information about those proteins, about how they work, what their structure of individual proteins are, all kinds of cool stuff related to that. And if there is a drug out there that affects that protein, it is going to update that as well. So it will contain information on what interacts, what molecules interact with your proteins, how they affect them, if they, activate them, suppress them, all that good stuff. In addition, so it has a place where you can analyze data. So if you all open a tab where you go to reactome.org, you can kind of look through this with me. Yes, these are the same people as the poster project. So from last week or the couple of weeks before, whatever your project groups are, this is the same group. You guys are going to do all the assignments from, other than the quizzes that are individual. All your main assignments at this point are group assignments doing with your group partner for your project that you're developing your poster with. Because remember, this thing that you're developing, all the work you'll do today is going to go to your poster in the end of the day. Okay, that's going to be a major figure in your poster at in the final. Okay, so this particular software or tool can do a lot of things, right? You can simply just browse pathways in here. For example, if you're interested in a particular gene or protein that you want to work on, you can just write that particular name. And so I actually clicked on it a little bit prematurely. You can just write that particular gene's name or that particular reaction, and you can just hit go, and you can get all the different reaction pathways that are associated with it, all the different reactions that are associated with it, right? So it's going to give you a lot of information, all the interactors with P53, all the complexes that it can find. And you can see, if you look at it this way, it's going to get overwhelming because there's a lot of information, right? And that's going to get really crazy. Just pathways alone, there are 64 different pathways that can affect P50, that P53 is somehow involved in. Lots of information. Now, you can also design, find specific, if you go to Reactome, you can actually look for, you'll have to, for this one, you have to kind of download this Reactome software, similar to how you did the ImageJ. Once you download it, you can look for specific pathways that are associated with a disease or a condition. So again, if you don't have any information at all, and this is where you're starting, you can say, I want to know if this particular drug has any known pathways that are affected by it. Or I want to know if what pathways are most affected in cancer, right? So I was trying to go back to Reactome. So here it also kind of tells you that it talks about patterns of pathways in different diseases and other things. In their documentation, they have a lot of information on how you browse this database and different tools for data analysis that you can use within it. So this is kind of a tutorial, you know, or how to book section of the platform. And then they have what we are going to use, which is the Analyze Data. Now, if you click on this Analyze Data tab, it will open up this window, and it will give you lots of different choices that you can use. Now, sometimes we are running an analysis where we already have a short list of genes and proteins that we want to focus on, and we maybe have some information about them in a normal condition versus a disease condition. And we have it maybe in an Excel file. Then we would just choose the file and upload that, or a text file. Usually it takes text file or Excel file. That's for when we have large data sets that we already have readily available, ready to use, and we want to put them in there. Now, you don't have to have a file ready to be put in here in the right format. You can actually put all kinds of different information in here as your starting input. You can put the protein's identification number, their unique ID numbers from NIH website. This is the way that I'm going to teach you how to use today, where if you have a small set of proteins that you are interested in, you find their unique protein identification codes, you add them into this software, and then you can run your analysis. Very simple, very clean, no issues. You can do the same thing using the gene name list. So let's say you don't have the information on the protein IDs, but you instead have a list of genes that you're focusing on. Well, then you can look up the gene name list and you can use the gene names. If each gene, just like a protein in the database, has a unique ID number as well. So if you don't have the gene name, some softwares or some articles that you look at may be providing you with a gene ID number instead. You can use those numbers instead to look up your pathways. So it gives you many, many different ways that you can look at the same, look for the same information at the end of the day. So if I have a list of 4 proteins, I can put them in as their protein identification numbers. I can put them in as gene names. I can put them in as gene ID numbers, any of those. In addition, sometimes I have a small molecule, like one of your drugs, right? You know, the active compounds. Each active compounds in the NCBI NIH database also has a unique ID number. You can look up that ID number for your molecule. Let's say it's one of those weird molecules that they don't have a lot of information on yet because it's brand new. There's not a lot of research out there. You can actually look up the active compound in NCBI. You can look up its ID number and just put that small molecule's ID number in here. And then look up what comes up, if any pathways come up through that. That's another way that you can find information on pathways associated with your compound or your natural product. And so these are just two different databases that contain all the names of molecules. So the KEGG and CHEVI, they're two different databases that are available internationally for small molecules. In addition, there's another thing you can do. So if you ran a gene expression array where you looked at all the genes present inside the cells with and without your drug treatment, for example, you can put in that entire data set. Usually it contains 40,000 transcripts in a text file stage, right? You can just put them in. So this one shows a control, and then after treatment with whatever, 10 hours, 14 hours, 18 and 24 hours, how the system changed. You can put all that data in and then have the software analyze it for you. Takes away from all the coding I used to do and would just do it for me and look up things that are affected or not affected. So that's a really, really powerful ability of this software. Same thing with metabolomics data. There are nowadays metabolomic arrays that look at a set of small molecules or a set of nutrients or a set of things that you're looking at, and you're looking at how they're changing in your different samples, different cell lines, different conditions, and how they're metabolized by in those settings. You can put that data in. You just need to be sure that you're specifying what data is you're putting in, because if it doesn't know what input is there, it won't know how to solve it. So like I said, any one of these can be used as your starting point. And then there are a couple of other stuff. So this is looking at cancer gene census, cosmic data from online, looking at different genes associated with cancers and how they're affected in somatic versus germline, and looking at that, or tissue specific effect. So for every single gene, obviously, if we look at any gene in our system, it's not going to be expressed at the same level in a brain cell, in a neuron versus a hepatocyte versus a retinal cell. It always is different. So looking at this, we can look at relative expression of genes or proteins in different tissues in a particular organism, and then compare it and look at how it is changing in there. Okay. But we are going to use the UniProt. That's the one that we are going to try to use because that's the simplest of the searches. Now, to start with, you're going to need a list of genes and proteins that you can use to input in here, right? So this is why, if you remember last week, I kept saying, make sure you're looking for basic science research papers with names of genes and proteins that are affected by a molecule or that are affected in your condition. So what you want to do is make a list of those genes and proteins that you found. Now, the only kind of caveat to this is that we can't use gene name for half of them and protein name for half of them. So we are going to find the protein ID numbers for all that, all the things in your list, and then use that as your starting input, right? Because all of them should be in the same kind of format before you put the list in here. So in my example, in the assignment, I have, for example, a list of proteins that we are doing, right? I have P53, BAX, cyclin B1, B21. So you are going to have your own set of nodes. These individual genes and proteins that you find from your articles are your starting input. They are your nodes that you're going to be working with. Now, for those nodes, it's important for you to know, for the purpose of your final schematic, if they were up or down regulated in your system, okay? So when you gave, for example, Taxol, was P53 activated or suppressed? Or was BAX protein upregulated or suppressed? So you wanna know that information and you wanna write that down in your list so you can make your final schematic appropriately. Any questions up till now? By the way. Cool. So you want to make a list of these as your step one. Now, You want to strive to get 5 nodes minimum. You don't want to use more than 10. It's going to probably get too complicated for you. So you want to go for, if you have one of those compounds or one of those products that have a lot of information out there, you want to pick the 5 or 10 that are most affected, that keep popping up in multiple papers. And if you are one of those who is struggling to find them, you may be only able to get 3 or 4, but you want to try to get to 5. And the way you can do that is by adding in genes or proteins. So if you're looking at your natural product and you only find 3 in there, you may want to go back and look at your disease or your condition and see what genes and proteins are affected by that condition, and then run one with those to kind of make up for not having enough nodes just affected by a natural product. But for most of you, 5 should be easy to get. 5 is, I'm giving you a small number as your minimum required. Okay, so you want to get those 5 nodes ready. And then what you will do is, I'm going to close this tab first. So you are going to go to this website called, it's still the NCBI that we've been working with, the National Library of Stuff by NIH. It's, and you are going to go to its OMIM kind of database within it. So once you go to NCBI, it will pop up this big long list, right? And you can select for OMIM through that and say, I want to look at OMIM. And OMIM is good because it is really comprehensive, right? It gives you a lot of information on the genes and proteins that every single thing that is published on, it has it. And it has all that information about them. So once you get there, you are going to then input the names of your nodes, right? So I'll put in P53, for example. And you want to make sure when things pop up, it's going to pop up a lot of stuff. And sometimes the first one is not going to be the right one, right? This one is long intergenic non-coding RNA P53 induced transcript. Well, that's not P53. It's something induced by P53. The second one is the correct one, tumor protein P53. You will click on this. And you want to then, next thing you want to do is get the protein ID number from it, right? So to get the protein ID number, on the right side, you will see this link of external links, right? This little line. Now, if you look in this has a lot of information about this protein. There is a lot of information about protein within the OMIM, but then on these external link, there are lots of links there to get to the information you want. For example, I could go and search the information about it in the genome in various species by going to the genome setting, either through the European databases or through the American databases. You can look at the DNA, and again, it will have links to several databases, international databases in there, both European and US, that you can go to those browsers to get DNA, actual DNA sequence of the gene and various species, including humans. You can have the protein information. And again, it has several databases it's connected to. You are going to go to UniProt. Okay. So it will open up a new place, and actually it kind of gives you the protein ID number right here, UniProt number right here as well. Oh, la, la, la. Internet, don't be slow. Of course, I don't have patience either, so it doesn't help. Oh, come on, there you go. Now, when it opens it up, you want to make sure that what you're using is the human P53. And you can also kind of specify it in the OMIM when you input it, just write P53 human. So it kind of gives you just the human versions of the stuff and takes away from any other organism that you may be getting data from. Okay, so this number is what you're looking for. Okay, that's your first number. Now you want to probably keep a document going where you can write these numbers, right? So you can just copy these And you just want to have a list of them, right, without any other extraneous stuff, because what you're going to input is just these numbers. And so I'm going to repeat this for a couple other proteins. I'm not going to do all 5. So we can have some numbers to get started with. Let's do BAX. Again, the first one is not the right one because it's talking about BAX inhibitor motif containing proteins. You want to read, don't just click on the very first thing that comes up. And like I said, that you can kind of make sure that you're getting your protein in humans by writing humans in front of it. So now you got the BAX protein as the first one. You're going to click on that. You're going to go to protein, and you're going to go to Unicron. And now it's going to load. So while it's loading, because it's taking its sweet time, let me show you a couple other things. If you look in this little bar, you actually see gene info, where you can get the NCBI gene number or the actual, the gene ID. You also get other stuff. You have clinical resources for different genes. You have the variations that are present that have been found, clinical variations that can cause diseases, for example, or that are just normal variations within the gene in different populations. You will have links to animal models that are associated with BAX mutations, whether it's in mouse or zebrafish or Drosophila. Those are going to be on this site as well. And then finally, you will have links to cellular pathways. Now you can get to 2 different, there are 2 main open source databases, platforms for cellular pathways, KEGJ and Reactome. The one that we are talking about is Reactome. So you could actually get to the Reactome pathway related to BAX from here as well. The problem with that is that you're not going to get just that portion light up. You wouldn't know exactly what part of BAX pathway is associated with your condition or your molecule simply by doing this, because it's going to provide you with an overview of that entire molecule, right? It's going to, like with P53, it's giving me a whole long list of reactions associated with it, pathways associated with it, interactions associated with it. It's going to be overwhelming. So that's why we're not going through oming to the cellular pathways, okay? Okay, so did I get my protein name? There we go. Again, that's my second one, and I'm gonna do one last one, and then we'll be ready. Since I'm just doing a demo, I'll be ready to then show you what it will look like. Now you could probably go through this too. Sometimes that gives you a lot of extraneous information, which is why I don't go to UniProt directly. But let's do cyclin B1 human. And it's going to give me the different ones. And actually, the first one is not the right one. The second one, the CCNB1, is the cyclin B1. The first one is, again, something that interacts with cyclin B1, but not the actual cyclin B1. And this is the protein ID number. So I'm going to take it. Oh, no, I just want the number. And I'm going to put that in here. And now I have my little list to start with. So you're going to copy your list once you have them, only the protein ID numbers, no extraneous information. And you are going to go here, and I'm going to just get rid of everything else and write my 3. So you're going to have just a little tiny list. And again, like I said, I want you to have a minimum of 5, no more than 10, I think is fine. And click on continue. Now you want to project to humans so that you remove any background noise and you don't have extra stuff associated with it. If you wanted to have interactions, there is an interaction one as well. And there is one where you can look at non-human equivalents. And then you simply click Analyze. And there you go. When you get there, you are going to get It runs relatively quickly. Well, we just had 3, but even when you have a lot of these in the list, even with like 20,000, it will run relatively quickly in a half an hour maximum. Um, but usually within a couple of minutes. Majority of the analyses that I've run recently with 500,000 transcripts or protein run within 2 to 5 minutes maximum. Okay, so I get my list. Now, what am I looking for, right? So what you're looking at are all the networks. So this is one way that we could have organized the networks, all the main networks inside your genome or inside the body. So obviously there could have been more, but these are the ones that keep coming up. So these are the ones that are divided up in. If you look at the central area, right, the signal transduction, gene transcription, disease, many times these always pop up, right? I just put a list of 3, so there wasn't a lot in there for it to pop up everything. But every single time, since every single protein we are putting in is somehow associated with the signaling pathway, signal transduction, always going to light up. Gene expression transcripts, always going to light up. Disease, many times will light up, especially if you're looking at a condition, it will light up. So when you are doing this, you want to stay away from these 3, if at all possible, right? Especially your signal transduction and gene expression, you don't want to go in there. Disease, maybe it is something very specific that you're looking for and that pops up, then you may want to use it. But otherwise, I would stay away from disease as well. So make sure that one of your networks is not single transduction, gene expression, and preferentially not disease either. The others are up for grabs. What are you going to choose? Well, that depends on what is lit up the most. So when I look at this, cell cycle pops up, right? That's lots of different parts of cell cycle network are popping up, very much affected by my little list. So that will be one of my nodes, one of my networks that I will put into my slide for schematic. Program cell death pops up. Again, something cool, something interesting, probably associated with, and is lit up quite a bit. Again, one branch of it. So something important probably. I will have that as my second network. For the third network, you may have to play around a little bit and think about what it is that you're looking at. So if I'm looking at cancer, for example, if my list was coming at looking at molecule affecting a cancer cell line or a cancer phenotype, then I may not really care about metabolism of proteins, for example, or may not care about this immune system. I will look at what is left in my other, because you're only showing me 3 networks. You're not showing me all the networks that are lighting up. And I will look for the most interesting one based on the problem I was researching. So cell response to external stimuli, yeah, maybe, or DNA repair. Sure, that seems interesting considering I'm looking at cancer. So I may want DNA repair as my third network. Again, that's a judgment call you will make based on your disease, your condition, and your natural product that you're looking at, and what response you're looking for. So the first one is always going to be the one that's most lit up. And possibly the second one too. So the ones that are most lit up, the first 2 are those. And then for your third, you want to play around and pick something that affects your condition, your natural product, and directly relates to it. And those are what you would be putting into your final pathway schematic, kind of like this. So when you make your slide, on one side, you will have a little bar that would show me your 4 or your 5 nodes or your 7 nodes or your 10 nodes, whatever you're using as your input. For each one of them, you will show them as either upregulated or downregulated based on research, based on what you found through your research. You would then put a horizontal bar on the top where you talk about your networks, the main networks that are associated with your nodes. And so this is where I wrote that. Now, in my case, I've also talked, shown them as either up or down regulated. And I will tell you where you will be able to make that judgment, whether it's up or down regulated. Just looking at this, if, only your upregulated ones are in a network, then obviously that one is going to somehow be upregulated. If only, if you have a downregulated node in a network, then it's at least part of it is going to be suppressed or downregulated. So that's what you will do. You'll have your networks and your nodes. Now going to the pathways. Where am I going to get this information, right? You're going to get this information through Reactome as your starting point. So let's start off with something simple. This one, there's only one branch lighting up. So that makes me feel a little bit, easier to understand. Right here too, actually, there's only one thing light up. So, but this is one of the ones that I'm going to look at. So if you click on this, it tells you what it is that it's associated with. This is a DNA double-strand break repair pathway. And on the left-hand side, you will see it pop up in this event hierarchy. Okay, so this is where you're going to start getting information about this. So in DNA repair, there are many, many, many pathways that could be affected. There could be base excision repair, damage bypass, damage reversal, nucleotide excision repair, mismatch repair, a lot of different pathways. There's one pathway out of those many different ways the DNA repair works that is affected in my little subset of genes by whatever molecular condition I'm looking at. That's DNA double-strand repair. That's the pathway you're going to be focusing on. You can click on it further. It will open it up right there, and it will show you exactly where it is that it is showing an effect. I may have double-clicked it too many times a little bit, but you can look at here and you can see where it is that it is getting affected. Yeah, I clicked one extra time, so it went a little too deep. But basically, it was P53 activated double-strand repair pathway. I know that only because it says P53 binding protein here. I wanted to go one step back. So it's showing that it was activated because it's overrepresented in here in my, now I'm trying to go back. Yes. So now if I go up here, it shows me that this is the gene that was affected, or the protein, the node that was affected. It was the P53 tetramer, and that would have led to activation of the DNA repair pathway. And this happens because when I click on this, it tells me why that would have happened specifically this way. Why did this specific pathway get activated? Now, P53 can get activated by a whole range of activators. It can get activated through many, many signaling molecules, many types of damages, many types of external stimuli. It could be radiation, it could be hypoxia, it could be nutritional deprivation, it could be anything, right? A lot of different things, not anything, but a lot of different things. Each one of those activators are going to activate it differently. They are activating through activation of different modulators, right? In this case, this is through ATM. It lights it up. When you click on this, it lights it up on this left side. And it also shows it up here. If you click on this, it will say ATM phosphorylates P53 to activate it. That leads to activation of the double-strand DNA repair pathway. So in your schematic, when you are actually making your schematic, let me open a brand new slide here, which I thought I opened. So in your schematic, when you're building it out, right, after you have put your nodes in the corner, not. So after you've put your nodes in the corner, I'm not putting them in yet. I'm just kind of showing you a general outline. And your 3 networks up top. So you can do Control D, and it duplicates it. the exact same thing. And then you can, nowadays the newer version of PowerPoints are really good. It helps you align them right here, but you can spread them out. And then when you start building your pathway, for now, because we are just looking at one of them for now, let's say that you were looking at that P53 activation. We'll talk about changing the colors and everything in a second. So the P53, when you search that pathway, it told you that it was activated by ATM. So I would say, write that in. I would say, okay, it would have been ATM activating P53. I'm going to make an arrow to show that it's activating it, right? Or phosphorylating it and then activating it. But these are then two proteins that I care about, right? P53 was the actual node that I was working with, and I knew it was upregulated, so I'm turning it red. You guys don't have to use red and blue or red and green. You can use whatever colors you want to show me up versus down. Just make sure you write in your little legend whether it was up or down, right? Same thing with the arrows. You can change the colors. You can do whatever you need. You can use different colors for up activating versus suppressing. You can use, typically if we are inhibiting, we make the arrow where the arrow is like this with a line going horizontally like that on it. That would be suppression or inhibition. You can make that yourself in-house, right? Like this. And once you make it and you like the shape, you could go to arrange and group it. And now it's grouped, so it can go together like that, right? And you can make it smaller, less big, whatever you need, and move it wherever you want. Right. But yeah, so that's, and then you know that it's activating the DNA repair pathway specifically. So you're not gonna just make an arrow and say DNA repair, right? You're gonna, or double strand DNA repair. You're actually gonna look up the double strand DNA repair in Reactome like we were looking at it. And you're going to add some of those genes and proteins and show that pathway, right? Show the modulators, show the response. So let me go back to it right here. This is the simpler version for the ATM activation, right? It's ATM. It's phosphorylating the P53, activating it. That's that portion of the pathway. This is the part that's working. That's the part you're going to write down. You're not going to write down all this extra stuff, the MDM2 kinase, all this grayed out stuff, not important, not putting it in. You're putting in the part that is important in your condition. Okay? Okay. Oh, go back to here. Go back to my DNA repair pathway. And it will show you that pathway more in detail. And then again, you don't need to put all this information. You're going to put in just the most basic of, basic pathway associated with it. Sometimes it is a lot more complicated here than you need. And if it is there, that's the one that I wanted. So what it is that you care about in here is that you can show double strand repair. You can show that P53, once activated by ATM, binds to this, and it fixes it by activating RAD51. So, or, there are two ways that it shows it can be fixed. So you are going to take a subset of this information and put that in there. This is as much information as I need in there from you. I don't need every single gene in every single modulator in the cascade. I just need the basic ones. So the problem was there was a DNA double strand break. that activated the p53 through the ATM activation, ATM activated p53, that translocated to the nucleus where it bound to the problem at hand, and caused a complex to occur that was then that recruited RAD51 to fix the DNA damage. And if it was a non-homologous end joining, it would be different proteins, the Q complex that forms to fix it. So that's the information that you would write that would draw out on that side of your pathway. Cool? Is that good up till here before we go back to our big picture to get a different area? Good. So then to go back, you just say show all, and it should go back to the original if it's in the mood to do so. Yeah, so you clicked on that and then you say, no, I want to see all of it. So there you go. Now we are back to where we started. Now in here, you see there are many, many pathways that are lit up. You only put like 3 genes, right? So look at how many different pathways those 3 genes are affecting. That right there shows you that there's a lot of crosstalk between different biological pathways, right? They're getting activated or suppressed by the same genes and proteins. So you can kind of walk around these yellow ones and kind of look at whether these seem interesting. This is something that seems related to your topic. So I look around to see what I might find. Ooh, that one was good. So this is G2M transition. That would be something interesting. I don't care about the polio-like kinase for now. There is the G1 to S transition. That might be interesting in our case, like in my case, right? If I'm looking at cancer. And then I look here, and this is nuclear pore complex disassembly, the envelope breakdown. Maybe a little bit too much information, too molecular for us, for what we are doing right now. Sister chromatid adhesion. But just know that these are actual pathways that are affected by those one of those 3 proteins that we were looking at. Look here, again, G12S DNA damage checkpoint. That would go probably with the DNA repair, wouldn't it? That would allow us to make a crosstalk, a connection between those 2 pathways in our final schematic. P53 dependent G12S damage checkpoint. Perfect. Let's click on this. Again, it will pop up right here. It will pop up right here, right there. Now look at this. DNA is damaged. It activates ATM. It activates P53. That then activated, we already know, in our case, we saw that it activated the DNA damage repair pathway. That was one of the things it did. But look here. It activates other genes that cause G12S arrest. So that would be your second. Within the cell cycle, the actual pathway that we are looking at are one of 2. There's apoptosis, there's also G1S arrest. For cell cycle, it would be this G12S arrest, right? And so in here, you would now start building out that portion of your thing, right? So in cell cycle, and underneath, you can write down what it is exactly that it's working with. So here it was, for example, DNA repair. And then underneath, you can write some information about what it is exactly, double strand repair pathway. That was the specific one activated there. You may want to make them a little bit smaller so they are not as bulky. And it looks nice and clean. I'll make it smaller. And so over here, what we saw was G1S or checkpoint, right? G12S and G2M checkpoint. And then also possibly apoptosis. Well, I'm going to leave the apoptosis out of here for the moment, and then I'll show you when we will put that in. And so now what we found out here was that this is also going through P53. So it's the same activator. So that means we shouldn't put that right here. We should put it where they can both show that crosstalk. And we can show that ATM activating led to the DNA repair pathway, right? And I'm not drawing them the nicest way right now because I'm just kind of demonstrating. And that would be your DNA repair pathway down here. And then when it's activated, and now you can go back and see what was activating it up here, that led to the G12S arrest. And that pathway will go right here. So what you want to make is a cohesive pathway, one schematic that shows all the different parts, all the different networks in one pretty picture, rather than three separate little mini pathways shown underneath. And that's an important thing that I want you to focus on. So right here, actually, it kind of already shows it a little bit, right? It shows ADM activating this and then causing arrest. And then it has an arrow going to apoptosis. And keep that in mind for a second because we are going to find that out in a second. And in our case, we also saw the double-strand DNA repair. So that would be one side of the story. This also shows you that there's a protein upstream of it, MDM2, that may also be playing in this activation of the G12S arrest or G1 arrest pathway. Okay? Okay, so now let's go back to our initial picture again and look at that last portion, right? We had a third network we were going to look for. So I'm going to go back here. No, I want to go back. I got you. There's lots of stuff changing there. I see that. There. Look here. Program cell death. That's my third pathway or network that I was going to look at. And that one has this one area lighting up. It has many branches that could be lit up. Again, apoptosis happens, you already know, through multiple mechanisms. So if I were to click on apoptosis, it will open up all the ways that it can happen on the left side, right? I can go down here. And apoptosis can happen through intrinsic pathway, through extrinsic pathway, through death receptors, through this thing, through that, right? Multiple ways. Only one of those branches is lighting up. Well, which one is it? It's the intrinsic pathway of apoptosis. So again, you can do the same exercise. You can look at exactly which protein is responsible out of those three that you put in. And it shows that it's the Bax protein. Now, if you remember from before, from our own learning throughout the semester, when we talked about caspases and intrinsic and extrinsic pathway, that also can go through P53. And it's up here as well. Cellular stress can induce P53, that can induce Bax protein, that can cause release of cytochrome C and all that good stuff. So you can actually see that third connection right there. So your P53 in my case is actually a central character in my schematic. It's going to be responsible for activation or suppression of all three of the networks that I was showing. Cell cycle is suppressed because it causes arrest, right? So that would be what that block would turn green. My DNA repair pathway is activated because the DNA repair pathway was activated by P53 in response to damage. And again, apoptosis is going to be activated because Bax was activated, and that would release cytochrome C, that would activate caspase 9, that would lead to death. So when you are making that final schematic, that's where you change the colors of these, right? So this is programmed cell death, right there, and Control T, we find out this is intrinsic apoptosis pathway. And now I would move this even more to here, in my schematic so that I can show, and maybe down a little bit because each one of these are going to be affected by different things, right? And then I will have my various pathways getting activated through the central controller mediator. So, at the end of the day, you're going to have your intrinsic pathway of apoptosis coming here through back. So it's going to be backs, and then your, Caspase-9 and all those stuff over here. You will have your check2 and all the proteins associated with the DNA repair pathway, double DNA repair pathway over here, right? Non-homologous versus homologous repair ones. Because we didn't have additional proteins, we don't know which one of those two would be there, so those two would be options. And then here you would have your G1 arrest pathway. Okay, and up here, obviously, like I said, we found MDM2. So all the ones that were not part of your nodes, you should keep either a gray or a blue, like whatever color, just some neutral color. And then the ones that are activated, make sure you tell me what that color will be. And ones that are suppressed, the nodes, actual nodes you're working with, those should be some colors that I can clearly see. Now here, after you do this, you realize which ones of these are going to be what color. In my case, since I'm using the red-green story, so cell cycle is suppressed, so it turns green. These two are activated, so they turn red. Okay? Questions? This chat has walked away. Sorry, 1 second. Let me bring the chat somehow. Well, what happened to it? Oh, I should find you, chat. Come back to me. There you go. Oh, it's not where I want it. Okay, so I wish it would move. It's not. Okay, there we go. I got it. Okay, so that's essentially what you're doing this week, is you're getting your, that you are getting your nodes that you need to create your pathway analysis. You're going to input those nodes after you change them into protein ID numbers into Reactome, and you are going to be working with that to get your individual networks first, and then within the networks, your individual pathways. Now, let's talk about where you might find this information about the nodes. Let's say that you're still a little bit struggling about where you can get these numbers, right? So obviously within your, many of your articles, you would have seen either Westons or other information right there that you can use. If there's a Weston there, it shows if it's upregulated, downregulated, or an ELISA showing the protein expression or activity. That's one place that you can get individual nodes. Some of your papers will actually have data sets, right? They will have genome arrays, and then they will show specific genes that were upregulated or downregulated. They would show them, verify them with RT-PCR, qPCR, some other method. And they may have a table like this. So here is an example of those. In this case, you will have multiple proteins or genes listed. You may not want to do all of them, right? You may want to just select the few that are related to your condition specifically. Obviously, you don't want to take 200 genes and work with them necessarily. A couple of the people have indeed found entire geodata sets, so like whole data sets of information that they can download and examine with or without Taxol or with or without another drug that they were working with. Obviously, nobody was working with Taxol. I just used that as an example. But NCBI, again, the same place that you get OMIM, in their choices, they also have this thing called geodata sets, or geo profiles, where you can put your search term for your disease, for your molecule, and pull up submitted data sets from other researchers. This is actual data from other researchers that you can use to do your own search. That's another place that you can get that information as well. So I just wanted to show you a couple of different places and ways that you may be getting these nodes from. Okay. Now, another thing is when you're making a schematic, you want to make it look nice, obviously, and cohesive, and make it look like one image. It shouldn't look. So maybe both of the partners are working on different parts of this pathway. It should, at the end of the day, look like it was done together as one, right? It shouldn't, I shouldn't be able to tell that there was one little portion done by somebody else because it clearly looks different. So there are ways that you can make it cohesive. Let me first show you some example student products from last semester. So that were good. These were like really nice or, you know, appropriate looking products. So this is one from one of the groups that were doing red yeast rice and their effects. They had their nodes. And now they did choose a weird color for downregulated because they put them as red, but all their nodes were upregulated or downregulated in their case. They had their networks listed up top, and their because they chose metabolism as one of their networks, it had multiple pathways within it that they could use. And so they have those clearly identified. And then they made their schematic. If you look at it, it's very nice and clean. They have it very organized so that you can see all the places there is crosstalk between the two networks. And you can see how each one of that network has the pathway clearly defined, right? So that's an example of a reasonable pathway. Now you will say, well, wow, this looks really fancy. This is all done in PowerPoint, by the way. I will, again, show you where you can get these fancy images, some of these fancy images to incorporate into your pathway, again, in the Reactome. So I will show you that in just a second. And then here is another student product that, again, is example of a good one. They did have some that were upregulated versus downregulated nodes. They have their three networks identified up top. They talk about what part of the network it is that they were focusing on based on their problem and their nodes that came up. And then they show the crosstalk between them. So it clearly shows that when these are getting activated, how this GLUT2 is affecting the metabolism and how the activation of glycolysis is affecting the insulin receptor and its activation and its response. So it is very clear what was shown in this pathway as well, and how, again, the different networks were talking to each other. Okay, so how do we make ours look fancy? Well, to make our looks fancy, we go back to Reactome. And this time, I'm going to go back out of this analysis space. So I'm going to go back to reactome.org. And now we are actually going to go to scroll down towards the bottom to their extra services. They have a place called Icon Library. It is super cool. In here, they have unique icons that you can download and incorporate into your schematic. And this includes arrows that talk about, you know, these are the typical way we describe, like I talked about arrows, inhibition versus activation, something that is multi-step process where you're not showing the many steps in between, some simple related process, some place where it's transforming one product into another. So they have different examples of arrows that you can download. You just simply click on it. It gives you choices on how you can download. Usually I would say PNG file, and then you can download that. Going back, it has cell types, different cell types you can use as an example in your system, apoptosis, dendritic, all that stuff. You can take those, viruses, bacteria, all of those will be there. They have cell elements. So you can search for a particular element, or you can just scroll their library and look for things. So they have like adherence junction, actin filament, things commonly used in these pathways. Apoptosome. I know some people are working with plaques in the brain and other places, amyloid fibers that they can make into plaques. I think there are actually plaques in here too. So they have a lot of different things, DNA, all kinds of molecules you can use within your system. Same thing with different proteins and different receptors. Um, you can look for your receptor if you want to look for death receptor or GCPR proteins. They will have certain ones that are commonly used in here that you can, insulin receptor, I believe, is in there as well. If you have something that's specific to a particular tissue or site in the body, they have a few of these as well. Not as elaborate as some of the others, but they have some of those as well. Proteins, they have many, many protein molecules that are listed in here that you can look for. So yeah, there's where the plaque was. I knew I'd seen the plaque. They have the amyloid precursor and the plaque. So many icons that you can use to enhance your schematic, make it look cool and unique. Now, one other thing is, let's say you want to show a membrane, right? So membranes are actually pretty easy. Cell membrane, right? Is pretty easy to make in here by yourself. You don't even need any outside source in there. So let me show you, for example, how you can make a simple lipid bilayer. So you would start off by making a little lipid molecule, circle, right? Let's make the circle, since we don't really want it to be too intrusive. And then you can use these wiggly lines to draw out the lipid legs, all right? I'm gonna make the second one. I didn't want it to be that like straight and nice and pretty, but whatever. There you go. You can then select it and group it, and now you have one lipid molecule. Now I can take that and I can do Control D to duplicate it, okay? And I can put it right next to the other. I can select both of them and Control D to duplicate both of them. And now I can put them next to each other. So I have my first layer of lipids, right? Let me do a couple more. I'm actually gonna go arrange group again, Control D. So now it's all four of them together. So that's my, if you just want to show one line of lipids, that's what it would be. If you want to show a lipid bilayer the way it normally is, let me group it again. Control, oh, not you, sorry, this one. Control D, and now I have my two layers, and you want to kind of offset them to show that one is ahead of the other. You can also make it better. Again, you could just simply ungroup it and then color them too, but I'm gonna make these slightly lighter to show the front versus the back. Right, and it depends on how you want to show them. Right there. And so you can make very easily your lipid bilayer. You can add your little transporter or a receptor in between, and then have, you know, again, once you've done it, you can group it so they're together. It's really went on there. And then I put a receptor in between, let's say, or a transporter, right? I can then move it front, back, whichever way I need to show it the way I want. You can hit Control and scroll left or right, you know, the left or right arrow to move it little micro amounts rather than moving them big time. I can make this. You can show them front versus back if you wanted to by, again, going home, going to arrange and saying send to back or move to forward, right? And that will like, maybe I'll do this. There you go. Because I did send to back, it's gonna, see, I did send to back for this one, so now it's in between those two layers. This one is not. So I can do, again, the same idea here. I can say send to back. And now when I do this, oh, I sent to back too much. And one forward, and then it should go back. Yeah. Okay. So that's what you would want to do. And now I have my little membrane with a receptor or a protein transporting channel hanging out in between, and I can group it. So you want to keep grouping things as you make them final to keep them contained. So when you move them around, you move the whole thing around and it doesn't get destroyed. And you don't have to worry about it changing. You can make it tiny, too tiny, right? You can make it big, however you want, and work with it. Any questions? I'm going to stop the share for a second. Questions about any of the stuff that I showed you? Cool. I guess there were a couple of other things in PowerPoint too. Let me share it back actually, 1 second. If you go down, you see there are arrows right here that you can use for flowcharts. Going further down, there are different shapes you can use to make your own molecules. Further down, there are these arrows that you can use to make like different parts. You can flip them, you can show them whichever, well, probably not this way, but there. You could show them to do different parts of the pathways. So there are many, many different types of, icons and symbols right there that you can use as well. You don't have to necessarily do anything fancy using Reactome, but they have stuff in there that you can use to enhance your stuff for sure. Okay, so, well, that's all that I've got for today. And the rest of the time, you guys are free to just go and work on your pathways and have fun with it. This is supposed to be, a learning experience, but also kind of a fun, creative experience. Before you guys run away, make sure that you have answered the attendance question, which I have not given you yet. So let's see. Name a pathway, a biological pathway that you're familiar with. And so once you answer the question, then you are free to go. So just write the name of a biological pathway.
Thank you. Have a good week.
You too. Take care.
Professor.
Yes.
Hey, so I had a couple of questions about our pathways.
Okay.
Let me go back to the ones that I found. So we're doing dengue fever and like the effects of papaya leaf, how it like alleviates it. It increases platelet count, but it's been talking about flavonoids. Are those proteins or what are?
No, that's the active compounds within papaya leaf probably that is helping with the platelet counts. So when you look at your basic science articles, have you seen proteins and genes described in there?
Not in any of the 5 references.
So remember that you're not limited to those 5 references. Those were just what your starting point was that you're doing the annotation for, right?
Yes,
so if none of those have it, you may want to go back and look for some that do contain that.
okay.
Right? Now, it may be that you would not find sources showing papaya leaf extract or papaya leaf gene expression related sources. But then in that case, one, you can look at dengue fever and what genes and pathways it activates, right? And then kind of try to extrapolate from there what you expect that papaya leaf would be affecting out of those genes and proteins based on what you find. Or you can look at flavonoids and see if any of the pathways flavonoids in general activate, how they affect the pathways associated with dengue fever.
Okay.
Okay. I'm doing a quick search to see if I can get you anything or get you started with anything. So if you do like dengue fever, biological pathways, you will find proteins and genes easily. Okay, so that's one thing that you can make a list of is pathways associated with dengue fever. And then you can go back and say papaya leaf extract, maybe, let's see, extract, mechanism of action, or so if you just put papaya, I did papaya leaf extract biological pathway, and one article popped up right away. And that talks about specific genes and proteins associated with the papaya leaf extract. Now, they're not going to be related to dengue fever, but these are genes and proteins that will be activated by papaya leaf extract when you ingest it. So what you can do is make a list of genes and proteins affected by dengue fever, right? Do a pathway analysis on that, and then refer to this reference and say, well, these were genes and proteins that we saw in a reference that were affected or activated or suppressed by papaya leaf extract, and see if you can fit them in that pathway. See if the same ones are popping up by dengue fever too. Does that make sense?
Yes, ma'am. Thank you. That's a big help.
No problem.
Professor, I have a quick question, if that's okay. Would it be possible for you to post this recording?
Yes,
like
I
earlier
will. Once
than
it
Thursday
becomes
or
available.
Friday?
Yes,
Okay.
I will go ahead and post this one today, actually, just because this is a something that we don't have class on Wednesday, and so those students will need extra stuff anyway. So I'll post it today. Don't worry.
Okay, perfect. Thank you.
No problem. Joshua and Lice, do you need help?
Just a quick question for the pathways. So you were saying drawing the different images, like using the different shapes.
Yes.
You can just shorten the spaces in between each of the images, like
Yeah. You can make them tiny or large as you need it, depending on how many molecules it is that you're showing and how many pathways you're showing and incorporating in there.
Okay, thank you.
You're welcome.
Have a great day.
You too.
Thanks.
Hi, I sent you an email. I was just wondering, were you able to get that?
Yes, I'm glad to hear you're back in US. Yeah, your topic is fine. You can go ahead and work on that.
Oh, okay, perfect. That was the question I wanted to know. Okay, so I'll just go ahead and work on it. So um it's really, I just want to make sure I'm doing this right. So really the purpose of this project is for me to find this topic and like give an informational based on a research paper that I found.
Well, you're looking for multiple articles, right, that are going to talk about your hypothesis, or it may or may not prove your hypothesis, but you're looking for research for your topic. You're going to look for some clinical studies that show it in actual humans. You need to look up a few basic science related papers that talk about it in cell culture or animal models. You specifically want to look for the same kind of techniques that we've been working with. So cell viability, you can look for activity of proteins, something maybe with DNA, or in your case, I think you would most likely find like Westons or ELISAs showing activity of proteins or expression of proteins. So you want to look for some articles like that. For the annotated bibliography, remember you had 5 articles that you were showing me with their annotation. One was a review article that has to be no more than 5 years old, because the whole point behind the review article is to give you an overview of the research as we know about it today. So if it's older than 5 years, it's going to be outdated. And then you want to find a mix of those clinical and basic science studies for the other 4. Okay.
Does it have to be based on the review article?
What?
Does it have to be based on the review article?
No.
No.
The review article is just your starting kind of overview of this field, and then you are finding other clinical and basic science studies that relate to it. Okay, so that's one set of your research that you're doing. The other side was that we were looking at what a general public, source of information is, just Google or infomercials or TV ads, and seeing how your product or your natural supplement, whether it's ginger or cinnamon or whatever, is portrayed in the media, in the news, and the, for general public, what type of things they say it's good for, and see if your research backs it up.
Okay, so I had emailed you like a topic and a hypothesis. Did that look?
Yeah, I want to say that it was related to ginger. I read it. It was fine. I saw it this morning.
Okay. Okay, I just want to make sure, like, from now on, before I submit stuff, I want to ask you, because
Yeah.
I'm finally able to be here and get stuff done. I don't want to lose any more points. I want to make sure I'm doing things done. And another thing is it possible for, well, obviously not now, whenever you have time, I wanted to like go back and complete.
Yeah, do you want to meet me later today in office hours between 3 to 4? Is that okay?
I have a class at 3 with biochem.
Okay, so we can meet it then, or we might be able to meet a little bit earlier. I have a meeting today at 1 o'clock, and I don't know when I'll be back from it. So I don't want to say that let's meet at 2 and then I'm not going to be back at 2, right?
Okay.
Maybe tomorrow after my class. So we finished around 11 today. So if we finished around 11 tomorrow, after that?
Yes.
Okay, so let's work on meeting tomorrow and going over the rest of your assignments then.
Yes, ma'am. Thank you.
Okay?
I forgot to type it.
No problem.
Bye. Thank you.
Bye. Victoria, are you there? I don't think she's there, Jen. I'm gonna stop the recording.